Okay. So I guess the obvious question to start off with then is, what is the extracellular matrix? And really, this is just the non-cellular component of tissues and other organically organized or living things. This is going to contain molecules. Most of this is going to be secreted by the cells themselves that are occurring around this, or sometimes within the extracellular matrix. There are sometimes a few cells occurring within there. And this does a few things. It provides the structural and biochemical support that's needed to the cells. Is that too bright coming through the window, or can you see okay? You're good? Can anyone not see okay? Okay. I'll keep it open because there's snow out there. Where do I put your chair? Over here. So it has several functions of the extracellular matrix, and really roughly we can divide them into structural functions and then also physiological functions. The structural functions include several different things. Number one, cell adhesion. We want cells to stick to each other and kind of stick into some order in a tissue, because you don't want them just kind of like a massive oozing stuff all around. You want them in some particular arrangement. You want that tissue also to kind of stay together. You want it to have some sort of strength where it doesn't fall apart easily. But with many tissues, you want them also to be flexible. If all parts of your body were rigid and stone-like, that wouldn't be ideal. Some of them can be, like your bones. You can be rigid and stone-like to some degree is okay, but other tissues not so much. The extracellular matrix also has a list or a few physiological functions as well. These include things like regulating cell-to-cell communication. Anytime that you have communication happening between cells, unless it's passing specifically through a tunnel between those places, it has to go through the extracellular or some sort of extracellular matrix. This includes lots of hormones and things like that. They have to pass through several different kinds of extracellular matrix before it gets to its final destination. Also, tissue morphogenesis and differentiation. Morphogenesis being how does the form and the structure of a tissue arise. And then differentiation, actually having cells becoming specialized specific cells in a tissue rather than just like the stem cell of that tissue. And both of these are kind of related to the final thing. One thing that extracellular matrix is very involved in is wound healing. You have to have specific kinds of extracellular components in place for wound healing to happen properly. So we can divide the extracellular matrix into a couple different parts. The first being the basement membrane. This is a sheet-like type of extracellular matrix that immediately lies underneath epithelial tissues and blood vessels. And this part of the extracellular matrix is really very organized. It is an organized mesh-like network of proteins and other acellular components, and can be divided up into three different layers. You have the lamina lucida. This is also, you have the lamina lucida and the lamina densa. Both of these are so named because under electron microscopy, the lamina lucida is actually loosened through electron microscopy. It's fairly clear, so to speak. And then the lamina densa is dense. It shows up dark, essentially, under electron microscopy. Both of these are part of a layer called the basal lamina. And then below those we have the lamina fibroreticularis, or sometimes just called the reticulated lamina. And we'll talk a little bit more about what each of these contain and do in a little bit. I want to talk a little bit more about extracellular matrices in general before I talk specifically about what's contained in each of these. The other type of extracellular matrix that we have is just known as the interstitial matrix. Unlike the basement membrane, this is not very organized. It's kind of loosely organized, has mostly just kind of random groups of stuff, and it tends to be either gel-like or fluid. Oh, it's really warm in there. It's a lecture from out here. So I'm going to give you an illustration of this. Right up here, is that showing up there?
Yeah.
Okay. So right up here, this is the epithelial layer of the tissue. Then immediately below that, you have this pink. This pink stuff right down here. This is the basement membrane. So this is the highly organized section of this. You also notice it's not just underlying these epithelials, but also kind of lining this blood vessel right here. And that is dense and highly organized. This isn't just kind of like a random mishmash of proteins and other stuff like that. It tends to be fairly solid, fairly rigid, and allows cells to anchor to it directly. But the remainder of the extracellular material is something like this, where you have mostly just kind of, you have specific types of proteins and specific ratios, but there's not like a highly ordered structure to it. It's just kind of loose. And you'll occasionally get a few cells in there that are helping maintain the extracellular matrix as well. Both of these are really kind of exaggerating and expanding. Most extracellular matrices aren't this dense compared to the cellular portion, but some are. What kind of leads us just to kind of an aside here is that connective tissues are tissues that tend to have a lot of extracellular matrix and don't have very many cells. A general name for cells that occur in close proximity or inside the extracellular matrix that help secrete extracellular matrix proteins and maintain the composition of that are known as fibroblasts. So to give you a couple examples, of connective tissue that tend to be more extracellular matrix than cellular components, you have things like tendons and ligaments. Bones have much more extracellular material than they have cellular component, and most of the extracellular material is mineralized, although there's a lot of organic component to that as well. Another thing would be blood. Blood plasma, you might think of that as like its own thing. Blood is a tissue, it's a specialized tissue. And the blood plasma is the extracellular matrix of that tissue. So let's go briefly through the major molecular components of the extracellular matrix. And really they can be divided down into three major organic molecular components. Actually, water is probably more prevalent than any one of these, but we'll put water aside for the moment. So we have fibrous proteins, proteins that tend to be long and stringy and fibrous, non-fibrous proteins, and proteoglycans. Let me go back here for a minute. Okay, so let's briefly go through each of these. First being of the fibrous proteins, let's talk about collagen. So collagen occurs in most extracellular matrices, not all of them. You don't have a lot of collagen, like for instance in your blood plasma. But in most extracellular matrices, collagen is a fairly prominent component, and it is a major structural component in those that it is occurring in. And the structure of collagen is similar to some things that we've seen in the past. You start with single polypeptide sequences, and those are known as alpha collagen. Now, the really interesting thing about alpha collagen is it follows a very regular and repeating amino acid structure. It tends to be a glycine, proline, and then something else. The X up here being just some other amino acid. So glycine, proline, some other amino acid. and then that repeated over and over and over again, or glycine something and hydroxyproline. So like glycine something, hydroxyproline, glycine something, hydroxyproline, on and on and on and on down the line. You might be thinking to yourself, what is hydroxyproline? We'll get back to that. Now, each of these alpha collagen strands then can be arranged, or are arranged, into a triple helix. So 3 of these strands helixed around each other, twisted around each other, and this makes a strand known as tropocollagen. And then several tropocollagen can be cross-linked together to make what's known as a collagen fibril. So we have all these 3-stranded, twisted, string-like structures, and then many of those together make up a collagen fibril. That would be cooler in there, but there's people doing stuff in there. Okay, never mind. I was an interloper. Don't like that feeling. Okay, anyhow, so here we have kind of a really brief kind of cartoon of that. We have up here in the upper left, this would be the alpha collagen, so different alpha collagen strings. And then those get twisted together to make a strand of tropocollagen. The process of that is called procollagen, sometimes as it's being constructed into the tropocollagen. And then many tropocollagen fibers then get put together into what's known as a collagen fibril. Now they're not laid out linearly like this, they're actually tubularly arranged, kind of a bundle. Let's see if I can draw something. Kind of looks like what it meant to look like. Anyways, so it wouldn't be laid flat like this. It'd be arranged into a cylindrical form like that. But they have a regular, right.
Doesn't this follow kind of the similar construction as the, I think it's microfibrils? Or is where they like start out as chains and then they produce like filaments?
Oh yeah, kind of like intermediate filaments where you have like the you have the strand-like monomers, and then they twist around. Yeah, so it's somewhat similar to that. You have very different amino acid sequences. Also, instead of just having a dimer, you have a triple helix rather than just a dimer, and then those are arranging in such a way. I mean, it's somewhat similar now that you have kind of an offsetting. And in this case, with the offsetting, you get this interesting arrangement here where you'll have a section in which they're not all there, and then where everything lines up. Does that make sense? And then another one where you get a gap between adjacent tropocollagen, and another gap between tropocollagen, but then an area where there is no gap. This gives collagen fibrils a solid or kind of a banded look to them. Kind of similar to what you would get with, it looks similar to striated muscle, but that being said, is at a lot smaller scale than that. Now, the structure of collagen is made possible because we have a couple weird, non-canonical amino acids that are necessary for this structure. And the first one being hydroxylysine, that's involved in cross-linking of tropocollagen together. And we also have hydroxyproline that got mentioned earlier. This is one of those repeating motif, so to speak, but it's not really a motif. This repeating structure uses hydroxyproline. And this facilitates really tight right turns that are necessary to get alpha collagen to create the triple helix with, what's it called? With 3 other alpha collagen strands. Triple collagen, wait. Oh, alpha collagen strands. What's that? Yeah. Did I say triple collagen? Okay. So this allows the 3 alpha collagen to turn tightly enough that it gets into a triple helix. If you did not have hydroxyproline, it's going to have a hard time making that structure. Now you might think to yourself, okay, how do we get these different amino acids in there? And actually, they start out life just as a lysine or a proline, but then you get post-translational modification happening in the RAR lumen. So once you get into the RAR lumen, you have lysyl hydroxylase converting lysine into hydroxylysine, or you have prolyl hydroxylase turning proline into hydroxyproline. Yes?
Are there more cases of these modified amino acids in the cell?
I do not know of them. No. There may be, but I do not. Hold on, let me think. I do not know. No. Once we now have several collagen fibrils, these generally get arranged into a larger structure which is known as a collagen fiber. At this point, collagen fibers are large enough that generally you can see them under a light microscope. So it would be somewhat arranged like this, where you have alpha collagen strands, 3 alpha collagen strands that get twisted together in the triple helix to make the tropomyosin or tropocollagen. I keep wanting to say tropomyosin because that's also something you have a lot of. But then those get organized together into a collagen fibril, into large bundles like this. And then lots of collagen fibrils get organized together into a collagen fiber. And then all the way along, you're getting kind of this banded pattern, and that becomes apparent also under electron microscopy when you look at a collagen fiber. So this collagen stuff is a major component of most extracellular matrices, not all, but most. And this is especially true with tendons, ligaments, and bones. They have a very, very high proportion of collagen in each of those. So just to give you a quick look here, I believe this is also, this is tunneling electron microscopy, and you can see this is individual fibrils of collagen, and you can see the banded pattern that you see here. That'd be like gaps and overlaps in the tropocollagen. And then this is demineralized bone. This is electron microscopy of demineralized bone, and all of these fibers, this is all collagen. So continuing on with the fibrous proteins, another one that you have quite a bit of in different places is called elastin. This tends to be in the extracellular matrices of tissues that need to be simultaneously strong but also elastic. You want them to hold together, but you want them also to be a little bit stretchy. So elastin is really rich in the individual monomers of elastin are very rich in hydrophobic residues. And this means that, like we've seen with a lot of other things rich in hydrophobic stuff, that means that it wants to ball up. It's going to be most stable if you can get all of those hydrophobic residues around each other and not exposed to the aqueous environment, so it wants to kind of ball up. And very strongly. And then what happens is you get many elastin monomers then cross-linked together, and this makes an elastic or elastin fiber. So visually this looks a little bit like this. When there's no tension on it, you have the individual monomers. They're tending to want to ball up and be kind of a more bunched up arrangement. You get this cross-linking between the monomers, but when you apply force to it, those will stretch out and become elongated. So once this is released, once the tension on this is released, it'll very quickly go back to the same conformation it had before. So elastin is important in places that you want to have, like you don't want them tissues to tear, but you also want them to be somewhat stretchy. This is places like your skin. You don't really want your skin to tear, that'd be bad. But you also don't want it to be really solid either. Blood vessels, your lungs, urinary bladder, other places such as that. So are there any questions real quick on the fibrous proteins? Okay, I'm going to skip over for a moment the non-fibrous proteins and come back to them. And I want to talk about the proteoglycans first. Proteoglycans, at their most simple, are just really, really heavily glycosylated proteins. When you typically have thought about a glycosylated protein, you probably think, hey, we have a protein. Is it okay if I erase OChem? You guys would be happy like not looking at that for a little while. Okay, no problem. So normally when we see a cartoon of glycosylated protein, it looks something like this. Hey, you have this protein, and hey, you got this little chain out there, you know, whatever. Hey, we have this protein. What's this little carbohydrate epithelium? That's not what proteoglycans are. This is like where the carbohydrates got unruly and ate the protein. They tend to be in the neighborhood of about 95% carbohydrate and 5% protein. And so these, they are proteins, but the carbohydrate component are these things called glycosaminoglycans, or just GAGs for short, surrounding a protein core. And glycosaminoglycans are these long, unbranched polysaccharides with a repeating disaccharide unit. There's a whole lot of different ones of these, and they will have these different repeating disaccharide units, but the sugars, the monomers that go into those disaccharides, that are then the repeating units, are specific special carbohydrates. First off, one is going to be an amino sugar. An amino sugar is just a normal sugar, but instead of one of the hydroxyl groups at one position, you have an amine group instead. And then a uronic sugar. Ironic sugar. A uronic sugar. A uronic sugar has, instead of just a normal, usually on the number 6 carbon, if it's a 6 carbon sugar, which most of these are, or 6 carbon sugar, the 6 carbon, it tends to have, or sometimes it could be other carbons. Instead of having just a hydroxyl group, it has a carboxylic acid. And again, these make up around 95% of a proteoglycan. The remaining 5% is some protein that those are going to be associated with that are in towards the core. And these are really diverse. There are tons of different proteoglycans that are making up the extracellular matrices, and these are going to be very different in very different extracellular matrices. This is one of the major components that makes different matrices act differently, especially physiologically. Let's go through and just look at a handful of these. Heparin sulfate, which is very in structure closely related to heparin, but they're not the same molecule. They're just very structurally similar, and they both occur in extracellular matrices. They're both proteoglycans. This is involved in things like angiogenesis. It helps to guide and determine the actual pathway of nascent blood vessels forming, and then also in blood coagulation. See, heparin is interesting because this is the proteoglycan that gets added to blood when it gets drawn to prevent it from coagulating. So if you take a blood sample, generally, and you need to store it for any more than like an hour to keep that from coagulating, heparin is added to it, and then you have heparinized blood that doesn't become a problem. Chondroitin sulfate is another one as well. You'll notice most of these are sulfate ions. And this is a major structural component of cartilage. It's also become a dietary supplement that you can go down to like Andes and buy. What's that?
Why?
Because some people think that if you eat this, it will help your joints. And the major point of this in cartilage, which is another connective tissue that has a lot of extracellular matrix, it resists compression. You also have keratin sulfate. There's actually large amounts of this in your cornea, kind of the clear area that's covering the surface and the lens of your eye. Now, if you have a cornea that lacks this can lead to macular cornea dystrophy. Essentially spots of translucent or opaqueness developing in your cornea. That's bad. Yes?
Is heparin the thing that they put in rat poison and makes them crush glass? Or is that something?
Oh, I think actually heparin is in rat poison because of anticoagulant. It makes you, if you get too much of it, you just start laying in control. It's bad. Okay, so let's now go back to the non-fibrous proteins just for a moment. And the first one I want to talk about here is fibronectin. Fibronectin is kind of interesting because it's kind of this, it's very gregarious. It likes to like shake hands with everyone. It's like the most popular person at the party that just like goes from person to person, or everyone just kind of attaches onto them. It's a dimer. It's bound then by two sulfide bonds between the two monomers. So on the C terminus, you have two cysteines, and they will then make a disulfide bond with the other monomer, and that's the only connection between the two dimers, which is really kind of unusual. This is really kind of a low amount of interaction you get between a dimer, but it makes sense because one of the main points of this, I'll come to in just a minute. So some of the major things is that fibronectin binds to a lot of things. The major things that it'll bind to is collagen, and then proteins called integrins that are in the cell surface. We're going to come back and talk just a bit more about integrins in just a few moments. But these are embedded on the cell surface, and fibronectin will bind to them, and then also bind to collagen. It actually binds to a lot of other things as well. This is part of the reason why it's good that it doesn't actually have a lot of connection with it between the two monomers. That allows it to have more interactions with other molecules. So here's kind of a a diagram of the binding sites on fibronectin. Here at the C-terminus end, you have the two disulfide bonds that be binding it to another monomer of fibronectin. Here is where it would bind to the integrin on the cell surface. You have a binding site here for collagen. Also, there's a binding site for fibrin. Fibrin is a clotting agent, so this is one of the things that is involved in clotting. But then you also have other areas that bind things such as heparin and other stuff as well. It will bind to other fibronectins. And this is just a few. Actually, you can get different fibronectins that have slightly different binding sites on them. And then here's kind of an example. You have integrins or integrins in the plasma membrane extruding out, and then you have fibronectin then binding to those. So this is one way in which cells can bind themselves to the extracellular matrix binding between fibronectin to their integrins, and then also to collagen or other things in the extracellular matrix. Another extracellular matrix protein that's really important are laminins. So laminin is a heterotrimer. So you have 3 different subunits put together to make it, and they're called, as often as when you have trimers, an alpha, a beta, and a gamma subunit, or alpha, beta, and gamma chains. And these they bind to each other to form a mesh-like net. But they also, like fibronectin, now bind to other extracellular matrix molecules such as collagen, and then will also bind to other cell surface components in the cell. So not specifically to integrin, but other cell surface components. So this is roughly what laminin looks like. You have your alpha chain that kind of forms the central chain, and then you have a gamma and a beta chain then that are kind of woven around that, making this helical region in the center. And then you have different regions. Like for instance here, there's a collagen binding center, there's an actin binding center here. Here's an integrin binding center, so I guess some of them do bind to integrins. Again, there are lots of different kinds of these. And you generally have different centers on different chains that bind to different things. So again, both of the non-fibrous proteins, just kind of in general that I've talked about, fibronectin and laminin, bind together lots of individual components, both in the cell and in the extracellular matrix. So let's go back just really briefly to the basement membrane. I just really briefly talked about those layers, but now that I've kind of given you some of the components of the extracellular matrix, I want to talk a little bit about what things are where in the extracellular matrix or in the basement membrane. So the lamina lucida, this is the part closest to the cell. We have a lot of laminin there. In fact, if you look up laminin, you'll generally the first thing talked about with laminin is that it's very prolific in the basement membrane. It is here mostly in the lamina lucida. And you also have a lot of, in the lamina lucida, integrin. Now integrin, most of the time it occurs in a cell membrane. It's occurring part of a plaque desmosome. Now if you remember to the cell junctions, when I was talking about desmosomes, I was talking about the belt variety. You have a belt desmosome, and then also plaque desmosomes as well. And I talked about them binding the cell not to another cell, but to the extracellular matrix. This is how they do it. They are integrated with, or they have integrin as part of that desmosome. And that is going to be interacting with fibronectin, and also laminin, to anchor that cell to the basement membrane. Beyond that, we have the lamina densa. The lamina densa is predominantly collagen. Now, one thing I didn't exactly mention with collagen as we're going through is there's actually lots of different kinds of collagen as well. The predominant forms that you have here are types 4 and type 6. And actually within each of these types 4 and type 6, there's a lot of those guys as well. Yeah, what was your question?
It says 7. So it is.
Oh, 7, my bad. Believe that over me. Yeah. Me and Roman numerals. And then in the lamina fibroreticularis, you have collagen 15. Just joking, that's 3. So you have collagen 3, and this is going to make up a network of collagen known as the reticulated fiber network. Hence the name of the thing. Okay. I got 10 minutes left, so let's talk a little bit about some diseases. Well, just time for one disease. Let's just do one disease. One disease that has to do with extracellular matrices. So let's first use scurvy. No one use scurvy. So scurvy is interesting. You probably don't have scurvy. That's probably good. It's really pretty easy to avoid. It's caused by a deficiency in vitamin C. Vitamin C is also ascorbic acid or L-ascorbic acid. And if you don't have enough, you get scurvy. The reason this happens is that vitamin C is a necessary cofactor for prolyl hydroxylase, an enzyme that we talked about a little while ago when we were talking about what component? Yeah, hydroxyprotease and collagen. Yeah. So, if you don't have much vitamin C, you can't operate your prolyl hydroxylase, which means that you can't make collagen very well. Now, most animals synthesize their own vitamin C. This seems like a really important thing. Why would you not make, if you need this so badly, why would you not just make it so that you don't have to worry about getting it from dying? And most animals do. So like your dog, you don't have to give your dog oranges so it doesn't keel over dead. It makes its own vitamin C. And it makes it have fairly common things. It makes it have glucose. What's that? I don't know. That's a really good question.
Pirates didn't know that.
Yeah, pirates didn't know about what's going on. They never did the empirical test to see if their dogs could have scurvy. Or maybe it was with a hint of irony. Pirates had a very subtle sense of humor like that, I'm sure. That's a good point. But your cat, how about this? Your cats also can't get scurvy. Horses can't get scurvy. Cows can't get scurvy. However, you can get scurvy because you don't make your own vitamin C. It actually turns out you have the gene to make the vitamin C. It's there. And it's known as the gulo gene, which stands for gulonolactone oxidase. But it's broken. You're actually missing a big chunk of it. There's 12 total axons in the gulo gene, and you're missing approximately 7 of them. So here's the pathway that goes about that. This is the normal pathway in most animals that goes about creating ascorbic acid. You start off with glucose 1-phosphate up here, and then beyond this, actual like glucose gets fed into this. But glucose 1-phosphate, it goes through a series of things. You get down to L-gluconate, gluonate, and then you make this into L-gluconolactone, and then you make this. This step right here is where the gulo gene is, the gulonolactone oxidase right here, and you end up with ascorbic acid. This is the gene that is not present in you. You can go through this entire pathway, and actually you can use the products of this to go on into the to make pentoses, and you're okay, and you're great. But you have the entire pathway except for this last little bit. And you're not alone, actually, so it looks like it's broken down several times in history. You can sympathize with most other anthropoid primates, yes.
Do we need to know the entire pathway?
No, you don't have to know the entire pathway. Mostly for your own just interest. So if you were hanging out with a marmoset one day, you could both commiserate about the need to get enough vitamin C so you don't get scurvy. Hopefully they don't have scurvy at the time I'm saying this. Interestingly enough, also guinea pigs, it seems to be broken, and in bats, it seems to be broken. It's broken the same way also in most anthropoid primates. So you and all the other great apes and all the other monkeys, you were missing these same exons. You're missing 7 of the 12 exons needed in there. So it's mostly, you have big chunks of the gene, but it's actually mostly been deleted and gone. Guinea pigs is a little bit different. They're only missing like 2.5. So they're doing a little bit better, but still like it doesn't work. Bats are even closer, but not quite. There's actually only a few small things that make it broken. Like, and we and guinea pigs, we don't even make the gene. Like there's not enough there to even like try to make the gene. Bats actually make guanylactone oxidase, but it doesn't work. Like it's there, it just doesn't do anything. So yeah, that's kind of sad. And actually, if you get outside mammals, it's broken quite a few times. Other places, teleost fish have broken gulo genes. A lot of birds do too. Anyways, those are breaking genes. So this causes weakening in your collagen. You can't get those nice sharp turns to make good, nice tropocollagen anymore, and it makes for weaker collagen if you cannot make your hydroxyproline. And this ends up causing weakening to places that really need collagen because you need that kind of rigidity and flexibility together to make these tissues not break apart. So for instance, blood vessels, this can lead to hemorrhaging, and hemorrhaging that doesn't actually repair itself very well. Skin breaks easily and does not heal very well. And bones and teeth also become more brittle because you are losing the strength in the organic component of those bones. And so left, which is the mineral component, it's actually the hydroxyapatite that is in your bone is really brittle just by itself without the organic component there. So other things like bones and teeth become brittle. And so bruising happens really easily because you get easily breaking blood vessels, easy hemorrhaging, just general weakness because you also have collagen and other things in muscle fibers that can get made, joint pain, teeth become loose and drop out. Just generally don't get scurvy. It's pretty easy to stop. So this, early on in scurvy, you get like a receding of the gum line here, but then late on, actually exposure of the roots of teeth. Again, a breakdown of the extracellular matrix in the gums. Early on, one of the early signs is a tongue that becomes inflamed and engorged, it's like larger, and also hemorrhaging that's having the tongue, so you can see those like bright, well, in the ear, not on here, but these are red spots throughout the tongue. So. And then later on, actually this is, I have another mention. This is actually really bad for early sailors. You probably have heard this before, but there's probably somewhere in the neighborhood between 1500 and 1800, somewhere in the neighborhood of like 2 million sailors that died just of scurvy alone, including on some extra long exploration voyages. The majority of the crew died of scurvy. So I think it was Magellan's voyage, he left with 240 some odd crew, and over 200 of them died of scurvy. So yeah, bad things. Okay, so with that, can't go into any more diseases, let's just skip to this. That is it. That is all of the material I will present to you this quarter. On Friday, we'll come back and we'll have a review of all this, but until then, there will be a quiz going up this evening that I've talked about. That's optional, but other than that, enjoy. What's that? The quiz is going to be over mostly stuff that goes in between last test and this test, but somewhat drawing on things that will be for that, but not a ton.
So I was going to ask if we had to know the process for scurvy. Were you talking? Do we need to know like?
You don't need to know the entire pathway. You need to know the concept behind it. Yeah. You need to know the concept. So you need to know that what's
Being affected in the process and what the outcome.
So I'd say these would be the big things. You should know why cause you're not getting enough ascorbic acid. That's a cofactor for prolyl hydroxylase. That without that you don't make very strong collagen.
So we
You should know that's because you were lacking a functional collagen that acts on the final step of the process.
You have to add the
Yeah, you should know that.
It's kind of an indicator that hey, we can take it by ourselves. We need to know that process, not necessarily that.
No, all the rest of it.
Yeah, that's what I figured you were saying. Like, we don't need to know.
Yeah, you don't need to know all the steps in that process, but you should know.
You should know the basics.
You
need ascorbic acid.
Yeah,
and then that works on the. I got
lactone oxidase.
Yeah. Are you gonna upload these slides soon?
Yes.
When is the quiz due?
The quiz will be due probably after the final, so like at the very end of finals week, probably. So like when, you'll have a long time.
My question is about the lecture recording. So, unfortunately, that's my favorite study tool. It cuts the heart.
I know. I'm sorry. I feel bad that it didn't.
Yeah, it's unfortunate. But my question is, so anything that you would have mentioned in class, because like starting from the textbook, I try to kind of do both. The textbook is like quite different from what you go over. I noticed obviously like your tests are all over like what you went over.
Yeah.
So, what would be like your recommendation for like how I could kind of go back and like pick up stuff that I might have missed in the lecture?
Maybe, okay, so looking at your notes, maybe grabbing someone else's notes as well and comparing what they got down compared to what you got down. So here's how I do it when I sit down and write tests. I'm going to open up my lecture slides and I'm going to start going through there and like looking at things and like thinking of questions. Now, the thing is I'm not looking at like my detailed notes. So it'll be things I remember I talked about, but so like the super fine details
May not. Like the collagen, for instance.
Oh yeah, I'll talk about it. Like that.
Yeah,
that was major. Yeah.
So if it's a general, so like if I go through all your slides and I found like general like overarching like concepts, that should, if I understand all of it, should that be like sufficient? Then?
That should be good. That should get you a good grade. Like I will have things in there that I said verbally that didn't necessarily make it onto a slide, but that's not.
You can kind of extrapolate based on the knowledge.
Yeah, if I understand it.
Yeah, if you understand it, it shouldn't be like unintuitive, and it won't be like there's so much of the things I just said verbally that it would like tank your grade totally. It might be like a few questions here or there, like like knowing this thing was important for that question, and I said that. But generally, when I say things, it won't be like just something I said offhand in passing. It'll be something like. I might not harp down for like 5 minutes or anything, but it won't be like something super minor that I just like said really in passing.
Just like
But like for instance here, I probably only talked about the collagen like in 2 or 3 sentences.
But it's a contributing factor as far as
that was. I felt like a fairly major part of that pathway.
I just need to know the names of those two. So I didn't get that little bit of that slide down with whether they were. That's why I was curious if you were just going to upload them.
Yeah, I'll upload them.
Okay. Because usually I go
and this lecture will be uploaded.
Yeah, I go through the process.